Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntec Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to quantify PFAS leaching and mobility at AFFF-Impacted sites. Our first speaker, Dr. Jen Guelfo from Texas Tech University, will talk about her research to develop a standard PFAS leaching assessment methodology by optimizing 3 standard leaching methods. Jen's presentation will be followed by a brief Q&A. Then Dr. Charles Schaefer from CDM Smith will discuss his research into the relationship between PFAS concentrations measured in soil and those measured in pore water, with the ultimate goal of facilitating site management activities. Charles' presentation will also be followed by a brief Q&A session, and we will conclude the webinar with a longer Q&A featuring both of today's speakers. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and provide it to you in your webinar registration confirmation email. If you cannot download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you're unable to view the slides because your screen freezes, try keying in Control and F5 to do a hard refresh of your browser. If you're accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to have difficulties, please call into the conference line shown here. You can also submit a comment using the chat box. Please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. And in case of continued difficulties, you can download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration confirmation email. Note that we will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. We do encourage you to get them in well in advance of the Q&A sessions. And we ask that when you submit your questions to please add your organization name at the end of your question so that we can identify it. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program area. Dr. Leeson has been with SERDP and ESTCP since 2001, and prior to this, she was a research leader at the Battelle Memorial Institute, where she conducted scientific research on in-situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for site remediation and industrial wastewater treatment. Andrea received her doctoral degree in environmental engineering from the Johns Hopkins University. Andrea, please proceed.
Thanks so much, Rula, and thanks everyone for joining us today. I am going to turn you over to our speakers today as soon as I can, but before I do that, I would like to give you a little bit of information about SERDP and ESTCP and just set the stage for the presentations that you'll hear today. I am the Deputy Director as well as the Environmental Restoration Program Manager for our 2 companion programs, SERDP, which is the Strategic Environmental Research and Development Program, and that's our really what I would consider a traditional research and development program. It is a partnership between the Department of Defense, the Department of Energy, as well as the Environmental Protection Agency. And what we're really doing there is to take fundamental up to applied research to impact real world management issues, environmental management issues. And then the companion program is ESTCP, the Environmental Security Technology Certification Program. And this is our demonstration validation program where we take the technology or the knowledge that comes out of SERDP or other research programs, and we then go to more real world scale demonstration and validations of the technologies. And the whole goal there is to transition that technology out of the lab and encourage broader adoption of the technologies and accelerate commercialization. We have a number of environmental drivers that dictate how we fund our work. First, we are looking at the sustainment of our testing and training ranges, as well as facilities and operations. And as you can imagine, that's a very broad overview of all the issues that could take place on these types of installations. Anything from maritime sustainability, dealing with threatened and endangered species, changing environments, unexploded ordnance, et cetera. It's a tremendously broad area. But then we're also looking at reducing current and future liability, and that can take the shape of a traditional pollution prevention, where we're looking at the elimination of different chemicals of concern or hazardous materials or processes that are in use. And then we also are looking at impacts from past practices. And this is a traditional treatment and management of impacted groundwater, soil, sediments with some of these emerging chemicals of concern. And that's what we're going to touch on today with our presentations. Our focus areas then fall into these 7 different categories. First and foremost is PFAS and AFFF, and our presentations today will be talking about technologies and issues within this area, but we're also looking at installation restoration and resilience, munitions remediation, sustainable energetics, corrosion and repair technologies, as well as electrical infrastructure and planning. And I show you here our SERDP and ESTCP PFAS efforts. You can find this graph of the SERDP and ESTCP PFAS efforts if you'd like more information on our website, which is serdp-estcp.org. And this is an interactive graph under the focus area of PFAS and AFFF. And you can click on any of these boxes to get more information about the various projects that were funded over the years on PFAS issues. For every project we fund, you will be able to find a project webpage about that project, which will give a little overview about the project. But then also for any documents that have come out of the project, such as final reports, guidances, et cetera, all of that gets posted on their website. And we also will show any publications, peer-reviewed publications that arose out of the work, and that's listed at the bottom. If you look in the chat room, the link to this page is provided there for you if you'd like to take a look. Key to everything we do is the technology transition of the knowledge and the technologies that we're working on. This takes a number of different formats, from developing guidances, manuals, videos, in-person training. But then one of the real keys is this webinar series to summarize the work that we are funding. Shown here are some of the upcoming webinars in the series. We put out roughly 2 webinars a month, and you can go to our website to register for all of these different webinars here. All of them as well in the past are archived. So if you've missed one, you'd like to take a look at them again, you can go here to take a look. If you go to this website, then you can see the upcoming ones, register for them, and then look at the archived ones as well. At this time, then, let me turn it back to you, Rula, to introduce our first speaker.
Thank you, Andrea. All right, well, we're going to go ahead and get started with Dr. Jen Guelfo, who is an assistant professor and the Edward and Linda Faculty Fellow in the Civil, Environmental, and Construction Engineering Department at Texas Tech University. Jen joined Texas Tech in 2018 following a postdoc at Brown University. For the past 13 years, her research has primarily focused on PFAS occurrence, state, and transport. In addition to her academic role, Jen also has a combination of consulting and industry experience, and she uses this background to inform policy and bridge gaps between research and practice. Jen has a bachelor's degree in geology from the College of Charleston, a master's in environmental science and engineering from the Colorado School of Mines, and a PhD in hydrologic science and engineering, also from the Colorado School of Mines. Jen, we're delighted to have you. Please go ahead and get us started.
Great. Well, thank you, Rula, for the introduction, and thanks to the webinar organizers for the invitation to speak to you today about some research that we've been working on related to integrated approaches for evaluating PFAS leaching and mobility. So a quick overview of this presentation. After this introduction, I'll give you an overview of a compendium of laboratory leaching methods known as LEAF. I'll show you some outcomes of the methods within that compendium of methods, show you how those can be used in a screening level evaluation, discuss some of our sort of beginning level inroads into comparison of this laboratory leaching data to field relevant conditions, discuss conclusions and benefits to DoD. So I wanted to start first by just introducing the overall objectives of this project. When we conceived of this project, we recognized that standardized tools were needed to assess PFAS leaching and mobility for activities such as site prioritization, fate and transport modeling, risk assessment, as well as evaluation of treatment techniques. And so this project consists of three primary pieces, and the first one is shown here, which is to adapt existing standardized US EPA leaching protocols for use with AFFF-impacted media. And then separately, we're also measuring absorption of a broad suite of PFAS and developing a predictive model of absorption. We won't get into these results in too much detail today, but the point here is to show you that both of these laboratory scale efforts will feed into an effort to compare laboratory results to leaching and mobility under field conditions. And when we put all of this together, our overarching goal is to develop a framework for evaluation and prediction of release of PFAS from AFFF-impacted media. So a quick overview of this compendium of leaching methods that I referred to earlier. It's known as LEAF. It stands for Leaching Evaluation Assessment Framework. And this is a set of standard US EPA methods in SW 846. They are numbered methods 1313 through 1316. And I'm going to tell you a little bit about each one of those methods here. And then as we go through data later in the presentation, I will remind you of the objective of the individual methods. So starting first over at the top left is method 1313. This is a batch test. It looks at liquid to solid partitioning, or LSP, as a function of pH. So in each set of batch reactors, you would adjust the pH to a different level between 2 and 13. Method 1314 over at the top right is a saturated upflow column test. It is intended to look at liquid solid partitioning as a function of liquid to solid ratio, or L/S ratio. Moving down to the bottom left-hand corner is method 1315, where you place a monolithic or compacted granular material into a tank of eluent, and we use that kind of tank diffusion test to evaluate mass transfer rates out of compacted materials. And then method 1316 on the bottom right is another batch test. And like the column test, it also evaluates liquid solid partitioning as a function of liquid to solid ratio, but it does it in a batch format. And so that may sound a little bit redundant with method 1314. It is, and we'll talk a little bit more about that when we get to the data. So you won't see data from method 1315 today. We will be looking primarily at data from 1313, 1314, and 1316. And so before we jump into that data, I just want to discuss a few of the advantages of LEAF, why we picked it for this project. The first is it is the standardized set of methods to evaluate source terms in the lab. And it can be used to bound leaching over a range of conditions. So it doesn't just look at a single condition like some other standard leaching protocols. You can look at a range of conditions, which also gives you the flexibility to focus on screening level or site-specific conditions, and also flexibility to evaluate impacts of varied conditions on source terms. So for example, if you want to know if a particular stabilization technique is going to reduce leaching, you could conduct LEAF on both stabilized and non-stabilized materials. So LEAF was originally published for use with inorganics in SW 846. So it is not published for use with PFAS. And so there were some optimization and validation needs centered around using these methods with PFAS. And they range from some of the more mundane aspects of method implementation and into some of the more maybe interesting aspects. And so these are just a few of the things that we had considered as part of this project. Analytical requirements, for example, and the sample volume needed to meet those analytical requirements, whether or not the materials of construction for the experimental apparatus are suitable, how to do mixing, but also especially physical separation for the batch tests. And then some of maybe the more interesting aspects of fate and transport that are maybe more unique to PFAS, like does there need to be an unsaturated column test or column protocol in order to effectively count for vadose transport. And then of course, because it's published for inorganics, it's also been validated. LEAF has also been validated with field data for inorganics. And so there also needs to be validation with field data for PFAS. So what you're going to be seeing today are data that is presented from the optimized methods. So we have implemented the 3 methods that I'll discuss today on 4 AFFF impacted soils. So this is just a quick slide to show you that those soils are from 4 separate DOD facilities in New Jersey, Virginia, and Texas. And so now I'm going to move into the method 1313 results. And I'm going to take a little time on this slide to sort of orient you to these plots because they will be similar on many of the slides that you'll see here going forward. So what you're looking at with method 1313, which again is looking at liquid solid partitioning as a function of pH, and it's using a batch conformation to do that. So we have eluate pH here on the x-axis. We have the constituent concentration on the y-axis, which in this case is PFPEA or perfluoropentanoate. And then you're looking at the eluate concentrations over the relevant pH range for 4 different AFFF impacted soils. And the original concentration in soil for each of those particular AFFF impacted media is shown here in the legend. So the other thing that you will see within these plots quite frequently is that one of these symbols has the bullseye around it. That is what we refer to as a natural pH indicator. So that means that this is the batch reactor in which there was no adjustment to the pH. So the pH is just simply that results from reacting the aqueous phase with the AFFF impacted soils. So within these method 1313 results, we saw 2 trends within the short and long chain perfluoroalkyl substances. So with the short chain, we saw a relatively flat liquid solid partitioning curve over the entire pH range considered, which is 2 to 13, indicative of high solubility and weak overall sorption. The long chain PFAS do show an increase in liquid solid partitioning with increased pH. This is something that's been seen in other studies. And the hypothesis there is that it could be attributable to 2 primary factors. One is electrostatic repulsion. So at increased pH, you're going to have increased net negative charge on many soils. And so you could have electrostatic repulsion between perfluoroalkyl acid anions and the soil. And the other thing that could possibly lead to mobilization here or increased eluate concentrations is the increase in DOC that occurs at elevated pH. And you're looking at that on this graph that's shown here now. The DOC concentration with pH. We can't necessarily separate out those mechanisms with the data that we have to date, although I will say that some studies have concluded that DOC mobilization is not necessarily a significant factor, but again, we can't separate those mechanisms here. Okay, so this is still the same graph of PFPEA concentration with pH, but we're adding now a graph that looks at, instead of concentration, the mass release in micrograms per kilogram of dry material over eluate to pH. And there are no data shown there yet, and that's intentional because I want to go over these ideas of total content and available content, which are represented by the 2 sets of dashed lines that you see there for each soil. So the long dashed lines are what we refer to as total content. This is basically release of the total mass present in the soil. It may be that there is a certain non-labile fraction in soil, something that's not available for leaching, and in which case there may be an available content. In this particular case, we are not seeing differences between the total and available content, but the available content is defined as the maximum mass released at pH 2, 9, or 13. And we saw that for perfluoroalkyl acids up to C8, so up to PFAS and up to PFNA, the total content is approximately equal to the available content. So going forward in these graphs, I'm not going to show both just in the interest of keeping the plots more simple. So now we are seeing that actual mass release data on the plot, and you can see that for the short chain PFAS, essentially at all pHs, the total content was available for leaching over the entire pH range. And then not surprisingly, because we did have a trend in the eluate concentrations with pH when we look at the mass release of something like PFOS, we see that it has an increase in mass release with increased pH. Although as we start to approach circumneutral pH, we're still seeing that mass release approach that total content. So now we're going to switch to the next method, which is method 1314. And it, as a reminder, is looking at liquid solid partitioning as a function of the liquid to solid ratio. And in this test, we are looking at that factor in saturated upflow column experiments. And so just like the previous plots, we have the eluate concentration plot here on the left, but now instead of looking at concentration versus pH, we have liquid to solid ratio on the x-axis. And on the right, we have mass release, again, versus liquid to solid ratio. And so we discussed what that natural pH indicator means. I want to pause here before we talk about trends in the data, just to tell you why that natural pH indicator is showing up here on the mass release for the column data. So the indicator is there, and it represents method 1313 mass release in the batch reactor where there was no pH adjustment, so at natural pH. All of method 1313 reactors use a liquid to solid ratio of 10. So basically what this natural pH point is doing is comparing the release in method 1313 at natural pH to the release in the column test at a liquid solid ratio of 10 as a consistency check. And we are seeing that the mass release at the liquid to solid ratio of 10 in the columns is very similar to what we saw at natural pH in the 1313 tests. So again, we saw two different kinds of trends in the perfluoroalkyl acid data. The first was the short chain perfluoroalkyl acids, where we saw a rapid initial elution. And then you'll see these gray lines on these graphs. This is something that's put typically on these leaf plots of the 1314 data, where it's showing what behavior would look like if it was solubility limited. We definitely don't see that kind of behavior here for the PFPEA. Moving to the long chain, so this is PFOS. Looking at the eluate concentrations, we can see that relative to the short chain perfluoroalkyl acids, there was a delayed elution, and there was some following of that solubility limited trend at the low liquid to solid ratios in the mass release plot. However, really what we're seeing is that for the PFAS that are the perfluoroalkyl acids that are up to C8, the release is limited by the total contents. And then I also just want to note again that the 1313 and 1314 results are consistent. And then if you'll note, there's more than one data set here on these graphs. That's because these methods have been implemented in replicates. And so the results that you're seeing here are highly replicable. And we'll show some results of replication of other methods here in just a few slides. So now looking at method 1316 results, keep in mind that the objective here is the same as 1314. So we're looking at liquid solid partitioning as a function of liquid to solid ratio, but this time we're doing that in batch reactors. And so the plots that you're seeing here are again for PFPEA, and the column data are still on them in red. So these you've already seen, but now we've added the method 1316 data in green. And so again, I'm going to orient you to these natural pH indicators. The red point is the method 1313 at natural pH that we already discussed. Now we also have a natural pH indicator shown in green. That is just a circle around the 1316 data point that was conducted at a liquid to solid ratio of 10. So 1316 is conducted with different liquid to solid ratios and different sets of batch reactors. And the bullseye is around the one with a liquid to solid ratio of 10. Because again, we can compare it to column release, but also at that point in 1313, it should be a duplicate point at that point in 1316, it should be a duplicate point of the 1313 point. So 1313 at natural pH and liquid to solid ratio of 10 is a duplicate of the 1316 point at a liquid to solid ratio of 10. So again, these indicators are just used to highlight the points where we should be seeing consistency amongst the methods. So the 1314 and 1316 results look pretty similar, and that's expected. Again, they're similar in intent. In terms of why you might have both methods, so 1316 and 1314, they generate very similar data, but 1314 is more robust. It's conducted under conditions of flow, whereas 1316 is cheaper and easier to implement. So you might, for project objective reasons or budget, want to select 1316 over 1314. The trends are similar, but they're not expected to track exactly. But if you do implement both methods, it does offer a consistency check, as we've seen here. So I said that I would show you additional results of replication, and that is what we're showing here. So we took these methods that had been optimized for PFAS, and they have been implemented in triplicate on 4 different AFFF soils for the tests that you've seen here. We already saw replication on 1314, so here I'm just showing you the results of 1313 replication on the top for PFOA, and 1316 data on the bottom for also for PFOA. So triplicate implementation of each of these methods that we've done so far. And so what we're seeing here is reasonable replication when we implement this in triplicate, keeping in mind that this has been done within a single laboratory. So we're seeing that the modified leaf methods for PFAS are reliable, and the data reflect consistent laboratory implementation of the modified protocols as well as consistent analysis. So ideally that would be conducted across multiple labs. That's not necessarily a part of this project because an inter-lab validation is a pretty large task, but it is something that is in the planning stages. So now that you have all this data, what can you do with it? And so to demonstrate that, I'm just going to use the data from PFOS from one AFFF impacted soil and just show you how you could use this in a really basic leaf screening method. This is in the technical documents for leaf. This is the screening protocol that they recommend for use. So this is something that maybe you would use to, for example, evaluate risk at different sites, or perhaps if you were evaluating a suite of constituents of concern using these methods and you wanted to know which ones were most likely to exceed regulatory criteria, you could use this sort of leaf screening evaluation data to do that sort of thing. And so the most conservative estimate of leaching, or in other words, the highest estimates of leaching concentration, according to the screening protocol, would occur if you just assumed that the total content of your constituent, in this case PFAS, that's present in soil, leaches into the initial liquid to solid ratio. So we're looking at eluate pH down here and PFAS concentration here. And under that scenario, the total content leaches into the first liquid to solid ratio. So it's just this constant high concentration across the entire pH range. If you wanted to refine that a little bit further, you could consider maybe only the available content, which again is the highest concentration measured in the 1313 test at 2, 9, or 13. Sometimes that may reduce your upper estimate of leaching. In this case, it does not, because we found that the total content was available for leaching. So that doesn't really offer any further refinement in this scenario, but it could. But that available content still is considering a very wide pH range. And so maybe you're not concerned with all of that entire pH range and you just want to focus on the natural pH. If we look at the maximum concentration that occurs at natural pH, so these dashed lines are showing you just around the natural pH range, and this horizontal line is the concentration that you measure there. If you assume that that entire concentration leaches into the initial liquid to solid ratio, now we see that there is a decrease in the upper estimate of maximum possible leaching for this particular soil. And then the last phase of this would be considering not just the relevant pH range, but also potentially the relevant liquid to solid ratio range. And for that, you could use either method 1314 or method 1316. Consideration of the liquid to solid range would be perhaps a site-specific consideration here if we just assume that we are mostly concerned about what happens in the first liquid to solid ratio. Even with that, we're seeing a reduction in the maximum estimate of leaching. According to the US EPA protocol, the one that you pick would be the maximum of these last two, but looking at it in this way shows you how the 1314 could further refine your maximum estimate of leaching. And from here, you can use these in various different screening evaluations. If you wanted to make these kinds of data more site-specific, that's when you're looking at things like infiltration rates to help you understand what the actual, for example, time frame is that's represented by this liquid to solid range down here on the method 1314 data. That would be one example of making this even more site-specific. So now I'm going to switch gears a little bit and look at this leaf data versus some of the batch sorption data that we've generated as part of this project. So on both of these graphs, and you're looking at log KOC values versus the constituents on the x-axis for the PFCAs on the left and the PFSAs on the right, or perfluoroalkyl sulfonates and perfluoroalkyl carboxylates. The blue data are method 1313 data at natural pH. So keep in mind, this means desorption off of AFFF-impacted soils at a liquid to solid ratio of 10. And then the pink data represent equilibrium sorption onto soil, also under a liquid to solid ratio of 10. But this time, instead of using AFFF-impacted soil, the PFAS standards are spiked into these unimpacted soils. And this was done for 22 unimpacted soils as part of this project. So each of the data points was normalized to the FOC for that particular soil. And so you're looking at the range measured for each of the constituents shown here. So a couple of takeaway points from this. First, especially with the sulfonates, but even to some degree with the carboxylates, we're seeing that the 1313 organic carbon normalized values at natural pH exceed those that we measure in the batch sorption KOC for many of the PFAS that are shown here. That is something that people have noticed in the past, that desorption off of AFFF-impacted soils that have been aged in the field is not necessarily fully captured by sorption onto clean soils that you would implement in the lab. And that suggests that an equilibrium KOC value, if you were to apply it to a site, may, especially in a source zone, may overestimate leaching of some PFAS. And we also see that, as with other studies, that factors besides organic carbon content likely matter here in terms of we tried to normalize sorption data to organic carbon content, but in reality, these data are suggesting, and other studies have shown, that other factors such as clay content and soil pH also matter. The other thing that these data suggest is that leaf with a pretty basic batch test with leaf with site soils may yield more representative source term assessments from source zones than use of the KOC values. Of course, this is the kind of thing that we have to put a question mark next to until we validate that with field data, which is something that we're working towards in this project. So we don't have any comparisons to field data as of yet. We have initiated looking at leaf versus some laboratory tests that may capture more field relevant conditions. And the results of that are what you are looking at here. So this is basically a first pass screening of the need for inclusion of an unsaturated column protocol as part of leaf. And so what you're looking at here are tests that were implemented, column tests that were implemented at 2 saturations so far, 100% saturated and then 60% saturated, which is still a pretty high degree of saturation. And we're trying to answer the question of, again, whether or not these unsaturated column tests are needed for representative source terms. And so what we're seeing here is that PFAS at 60% saturation and normalized to the liquid to solid ratio don't really have any systematic differences with the 100% saturation. And any differences that occur, for example, down here at the low concentrations, not only are they not systematic, but they're pretty small differences in concentrations that may be within analytical variability. We see that here for these 3 carboxylates, and we see similar results here for these 3 sulfonates. So evaluation of other PFAS as well as additional soils and definitely lower degrees of saturation is ongoing, as well as assessment of variably saturated conditions to see how well leaf data can capture variably saturated conditions, for example, rainfall columns. And we will also be comparing leaf data to field lysimeter data later in this year. And so with that, a couple of key conclusions from this data so far. First, that the perfluoroalkyl acid total content is available for leaching through PFAS up to C8. We are still evaluating the longer chain data because we're using field collected soils. Those longer chain compounds aren't present at as high a concentrations as some of the shorter chain ones. And so we want to make absolutely sure that we have our total content of these soils, that we're very confident in that. But preliminary data suggests that there could be some differences between total and available content for PFAS that are longer. We saw in the screening level approach that total or available content for the PFAS shown here as the most conservative screening approach for PFAS leaching. This likely leads to pretty high overestimates of actual leaching at a site, anywhere from 1 order of magnitude or more. And even within this screening assessment, by the time we got to the method 1314 data, we had decreased the estimate of maximum leaching by an order of magnitude. The modified leaf methods are reliable and replicable, as we've seen from the comparison of the replicate implementation of these tests. And the preliminary data suggests that the PFAS liquid solid partitioning, at least at 60 and 100% saturation, as a function of liquid to solid ratio, is similar. to identical. Benefits of this project to DoD is that at the end of the day, we hope this will yield standardized laboratory approach to evaluate PFAS leaching across multiple sites, multiple scenarios. The LEAF method, compendium method, offers increased flexibility relative to other methods that you may be more familiar with, like TCLIP and SPLP, which capture only a single condition. And so ideally, this can be more representative of the actual leaching that you see in the field. This also generates laboratory data that you can use in more in-depth lab field comparisons across multiple sites. And this work is all being done in close coordination with EPA, ORD, RCRA, and SERDP through my colleagues at Vanderbilt, who spearhead much of this LEAF work, and that should facilitate translation of these methods across different stakeholder groups. These are some of the folks who have helped me out in this work. I'd especially like to thank Isrek Riel and Fengfei Liu, who are the PhD students who worked so hard to generate much of the data that you see here today, as well as my other colleagues at Vanderbilt, CSIRO, University of Adelaide, and CDM Smith. Some of the resources that I cited within this presentation are here for your convenience. And if anybody has questions after today's webinar, I'd be happy to take those. My contact information is shown here, and thank you.
Thank you so much, Jen, for a great presentation. As a reminder for our audience, please submit your questions by using the Q&A box on your screen. Jen, we've received several questions for you. We're going to start with one from the North Carolina Department of Environmental Quality. In the pH batch studies, how long were the soils aged after the spike prior to elution?
So the method 1313 test is not a spiked test. All of the LEAF methods are intended for implementation on actual field collected soils. So I don't know exactly how long it's been since the last AFFF application at the 4 sites that we used in this study. I'd have to do some digging to come up with those numbers, but oftentimes we're talking about, order of magnitude of years to even a decade or more since the last AFFF application. So that's a pretty significant period of aging.
Thank you, Jen. This next question is from the Massachusetts Department of Environmental Protection. You showed PFPA graphs on slide 39, but appear to be generalizing these results to other short chain PFAS. Did you do experiments and generate data for other short chain PFAS?
Yes. So we're using the AFFF-impacted site soils for all of these experiments. So we're monitoring whatever PFAS are present within the conventional analytical methods for release from these soils. So it would vary from site to site based on what occurs in the site soils, but we do monitor whatever we find in the site soils.
Great, thank you. This next question is from the U.S. Army Corps of Engineers. On slide 30, could the observation of LSP increasing with increased pH for long chain PFAS just be an effect of desorption kinetics?
So we've done some kinetic tests to ensure that our period of equilibration for these batch reactors is sufficient for reaching equilibrium. But each of the reactors at each of the pH points was reacted as part of the batch sorption test for the same period of time. So the data from the 1313 concentrations they're looking at release over pH, not release over time. So I wouldn't expect that to be a factor here.
Great, thank you. A follow-up question from the Army Corps of Engineers. On slide 45, wouldn't the LS ratio in groundwater be considered infinite? And how do you translate this batch reactor results to actual environmental conditions?
So liquid to solid ratio, it could be infinite, yes, but let's say you're looking at a site and you're saying, okay, I'm looking at the time period since the last AFFF application, and it's been 20 years since that last AFFF application, you can use, for example, rainfall rates and soil porosity and other characteristics to estimate the liquid to solid ratios that have infiltrated through the soil at a given site in your time frame of interest, whatever that may be. I'm just giving one example. And so you can use both the 1314 and 1316, which is a batch test, in that capacity. The 1313 test, it is meant to help you understand sort of within the total content of the soil what fraction of that mass might be available, and it could also help you evaluate leaching under varied conditions. So if you're going to implement, for example, a treatment test that's going to result in some alteration of pH, you might have some pH dependent changes in the mobility of your compound, and this test allows you the flexibility to evaluate that.
Great, thank you. This next question is again from the North Carolina Department of Environmental Quality. Do you have any plans to evaluate sorption dynamics for straight versus branched isomers?
No, we are not looking at different isomers as part of this project. One thing we are doing that we didn't include in today's presentation is we are looking at sorption and desorption of a really large suite of what I would refer to as suspect PFAS. So those that you don't really have analytical standards for. We're using high resolution mass spec techniques to study sorption of those PFAS on site soils as well as in systems spiked with AFFF. And so through that, there may be some evaluation of different isomers, but we're not targeting, for example, straight versus branched perfluoroalkyl acids.
Thank you, Jen. And this next question has to do with potential applications of your results. Might they apply to biosolids?
So that's a great question. And there's, I believe, a lot of folks within US EPA that Vanderbilt's been interfacing with that have asked that exact same question. And so not necessarily as part of this project, but as part of, potentially as part of separate efforts, the methods, modified methods will be implemented on biosolids, but that's not something that's been done to date.
Thank you. This next question is from the Navy Research Lab. What methods are you using to determine the total amount of PFAS present in the samples that you analyze?
So the total content is the total content of each individual analyte. So if you're doing PFAS, it's the PFAS in the soil, total PFAS in the soil. If you're doing PFOA, it's the total PFOA in the soil, just to make sure there's no confusion that we're looking at a total PFAS number. So we're just doing the individual concentrations of each PFAS using the standard basic methanol extraction and mass spec analysis. We do have some tools for trying to look at total PFAS mass balance in our lab related to use of total oxidizable precursor and combustion ion chromatography techniques, but I just wanted to clarify that here that total content refers to the total content of an individual analyte.
Great, thank you. We have time for maybe one or two questions, depending on how fast you can answer them, Jen. Do you think, this is from EPA, do you think the estimate of available content derived from batch liquid solid equilibration is a reliable number or related to leachable concentration for both short and long chain PFAS? So
what we're seeing here, and what I've seen in other studies as well, is that there's just not much pH dependence to the short chain PFAS leaching and mobility. So to that end, yes, I mean, you can do the evaluation over a range of pHs, but we've seen in multiple studies now, at least in our work, that we're not seeing much changes in the short chain PFAS mobility at any pH. So I think those are mostly just going to be fully leachable. With the long chain PFAS, I think that use of the method 1313 test as to bracket the maximum possible leaching at pH is a valid approach. But of course, if you were just using that alone, we do still, and you did have an available content, for example, that was less than a total content, that would still be a very upper end of leaching and is likely to be greater than what you see in the field due to various various other field specific factors. So, but it's as an estimate of a maximum available content. Yes, I think that's a valid tool.
Wonderful. And one last question from UC Berkeley. How does the method 13 or 1316 desorption data look for precursor compounds? For example, zwitterionic precursors. Do these compounds behave like long chain compounds?
So that's not something we fully evaluated within this data yet. We really want to do that using suspect screening techniques so that we can look at the full suite of PFAS that are present in the soil. We have characterized these soils using the suspect screening high resolution mass spec analysis to understand more about all of the PFAS that are present here. And we have archived all of these samples. We've run them all just with targeted analysis so far, and to archive all these samples with the future objective of going back and at least analyzing a subset of these data for those broad suite of precursors. So I can't speak to that from this particular data that we saw here today, but in a separate aspect of this project where we are looking at sorption of a large suite of PFAS using AFFF spiked into batch reactors, we are seeing some differences based on charge, and it really depends on the type of zwitterion you have. So if that zwitterion is net negative in charge because it has 2 negatives and a cationic charge, that makes a difference relative to something that might be net neutral in charge, 1 anion and 1 zwitterion. And the position of the charge in our preliminary data, like for example, if you have a terminal anionic charge versus one that's central, also seems to make a difference.
All right, and we're going to conclude with this last question from Nnamdi Azikiwe University in Nigeria. Apart from the effect of pH on the mobility of PFAS in soil, can you summarize the factors that most impact the leaching rate?
So within this data, it seems like the leaching is going to be limited by the total content, but that's a very big question in terms of actual field processes that will influence fate and transport, some of which I'm sure Charles will address in the next presentation, because we haven't necessarily done the comparison of our lab data to the field data to know what some of the fate and transport mechanisms that folks have studied are most key to implement in a lab setting. But, you know, for example, one thing that might need to be considered is air-water interfacial behavior of PFAS, which is what we were looking at with the saturated versus the unsaturated columns. At 60% saturation, it didn't make a difference. At 30% saturation, it might, we still don't know. And so those kinds of field factors and understanding which are most important to capturing field relevant data is something that might ultimately not be fully pieced out until we're able to complete our comparison to the lysimeter data later this year.
Great. Well, thank you so much, Jen, for taking the time to answer these questions. We'll try to get to other ones towards the end if we have time. Otherwise, I would encourage all of our audience members to reach out to Jen at the information included in her last slide. We're going to go ahead and transition to Dr. Charles Schaefer's presentation. Charles is the director of CDM Smith's Research and Testing Lab in Bellevue, Washington. He has over 25 years of experience in evaluating the fate, transport, and treatment of organic contaminants in water, soil, and fractured rock. Charles has served as a PI or co-PI for research projects funded by SERDP, ESTCP, the Air Force Civil Engineering Center, the Navy Environmental Sustainability Development to Integration Program, and the Water Research Foundation. Most notably, Charles has been awarded the SERDP ESTCP Project of the Year Award 3 times, either as a PI or co-PI. He received his undergraduate and doctoral degrees in chemical and biochemical engineering from Rutgers University. Charles, please go ahead.
Thank you very much, Rula, and I am really thankful for this opportunity to present today. I'm going to be talking about an ongoing ESTCP project that I'm leading that's looking at PFAS leaching at multiple sites. So just in terms of an outline, I'm quickly going to go through some project objectives and the approach and methodology. Then I'll talk about results, first looking at some results from unsaturated soils and then some PFAS pore water concentrations, and then touch a little bit on PFAS equilibrium and perhaps and some phase distribution, and then wrap up with some conclusions and DoD benefits. So in terms of overall objectives, the overall objective of this project is to get improved insight into PFAS leaching from unsaturated soil. And we want to evaluate methods that we're using for measuring PFAS pore water concentrations and relate them perhaps to site conditions on PFAS leaching, and then at least to some extent evaluate the role of PFAS absorption at the air-water interface on these processes. So really what catalyzed some of this work was a previous project that we're in the final phases of, and this is a SERDP project that we were doing at Lakehurst, New Jersey, where we really did a very deep dive, a very detailed analysis of an AFFF site. And we had, as you can see in the figure on the left there, about a 14-foot by 14-foot test area that was highly instrumented. And I need to give some acknowledgement to the folks at AFFF, Dave Lippengott and Greg Lavornia, who helped to install this system. And we did a very detailed analysis here. And a couple of the findings we found from this site is we had really good repeatability among our lysimeter results. And again, we had multiple lysimeters, multiple depths. And you can see that the repeatability was very good there in the upper right-hand corner. We also found that the pore water concentrations were very reasonably described by equilibrium partitioning models, soil water, and even improved if we include absorption to the air-water interface. But the question we have is, do these findings hold true for other sites? Were we just kind of fortuitous here in that we got these results, or are these types of results that one might expect at many other AFFF impacted sites? And that really leads us into the current project where we hope to answer some of these questions. So our approach for this project was to look at PFAS soil and pore water data at a variety of sites, and we've been out to 6 sites so far: Peterson Air Force Base, Air Force Academy, Randolph Air Force Base in Texas, Jacksonville down in Florida, and also two East Coast sites, which I'm just turning East Coast 1 and East Coast 2. And at each of these sites, we looked at a very small area, about a 3-foot by 3-foot area, where we would collect some soil cores. We would install three porous cup suction lysimeters and then perform up to 3 rounds of pore water sampling there. We did high resolution soil sampling and PFAS analysis, both target and suspect. That was performed by Chris Higgins' group at the Colorado School of Mines. In addition to this field testing, we did some parallel bench scale testing, looking at batch desorption testing of these AFFF impacted soils. And we also collected some soil, and we did some pore water sampling of these soils in the laboratory to compare to what we saw out in the field. So just very briefly touching on some of the methods. So our field pore water methods, we used a very traditional porous cup suction lysimeters, and you can see the figures on the left to show how they were installed. And moisture levels, we would need to pull that vacuum for maybe several hours to many times overnight. On the right, we can see what we term the mini lysimeters that we use for our bench scale methods, and those lysimeters were obviously much smaller, but the porous section had about the same micron opening, which was somewhere around one micron. So ideally, we would use intact cores for this, and then pull a vacuum on those micro lysimeters, which is what you see in the lower right hand corner. However, for some soils, they just weren't stable enough, or they were a little too dry, and we actually had to wet and repack. And part of how we viewed these bench scale soil cores, we saw this as more of an equilibrated value than what we might get in the field. In the field, there might be a lot of transient flow happening, whereas in the lab, these cores were basically stable for several days to several weeks before we collected a pore water sample. So we considered the bench scale value more of an equilibrium value, in the field as maybe more of a transient value. And we'll talk a little bit later about how some of those results compare. So let's get into some of the results now. Let's start with the soils. And a couple of the sites we went to, Air Force Academy and Peterson, they were in zones classified as semi-arid based on the expected rainfall and amount of recharge. And what I have shown for each of these, and I'll just talk about the Air Force Academy on the left, are the some select PFAS concentrations as a function of depth. And we just have some perfluorinated sulfonates, PFBS, PFH-XS, and PFOS to kind of compare. And what's interesting is we see this kind of nice chromatographic effect. So the PFBS, we see its maximum concentrations down around 7 feet. PFH-XS, that center of mass, seems to be more around 4 feet, and PFOS around 1 foot. And kind of qualitatively the same types of trends for Peterson. This is very interesting and consistent actually with some results for other semi-arid results. Ilka Wallace recently presented or published a paper that showed something similar to this. I also want to point out for PFOS, at least I showed the apparent KD values for each of these sites. So you can see it varies by about a factor of 10 between these two sites. So again, this is very interesting to see this type of chromatographic separation. Now, in comparison, let's look at a couple of other sites, and basically all the other sites we've looked at so far are in non-arid regions, so a lot more rainfall. I'm showing just 2 sites for comparison purposes, but what we see here is a little bit of a different profile. We don't see that chromatographic separation, meaning if you look at the Randolph on the left, you see PFBS, PFH-XS, and PFOS basically are all showing that the same shaped profile and having a maximum concentration of around 2 feet. The KD range for these soils is about in the same range. And I think this is interesting because I think this might really highlight the importance of hydraulics for what's happening with PFAS migration and what the concentrations are in soil. So these hydraulics, recharge, things like evapotranspiration might be playing a very big role, and maybe at least to some extent, this is still somewhat understudied for PFAS at a number of sites. So let's jump into some of the pore water data we collected, and I have shown here just data from 4 of the sites we were at, where we have the PFAS pore water concentrations in micrograms per liter for a number of the identified compounds. Also shown are the number of samples that we have. Now, in some cases, we had a little bit less data than what we were happy with, and this is just because for some of the sites, the soil was dry, and pulling out sufficient samples for analysis was a little bit challenging. But I think overall, when we look at these data, and again, these are plotted on a logarithmic scale, and they're always a little more forgiving when it comes to looking at error bars. What we see is I think we have generally reasonable repeatability among samples within a factor of 2 or so. So I think that's encouraging and probably sets some sort of level of expectation. The other thing that we've noticed, and it's not just these 4 sites here, but at least another 3 or 4 that we've looked at, is that we're generally finding, say, for example, PFOS concentrations in a relatively, within maybe a 1 to 2 order of magnitude range. Of course, we're looking at sites where it's generally been 10, 20 plus years since they've been exposed to AFFF, but we haven't seen a huge amount of variability in terms of our PFOS concentrations. But I admit that could be in part because we haven't looked at dozens of sites yet. So looking a little bit more at what we're seeing in terms of the nature of the PFAS in these pore waters, so I have some figures here showing on a percent fluorine basis what the distributions were between perfluoroalkyl acids in red, target or quantified precursors, and also suspect or semi-quantitative precursors. And we saw maybe somewhat surprisingly some big differences here. You know, for example, Peterson and Jacksonville were really primarily dominated by PFAAs, and that was a lot of PFH excess, a lot of PFOS, whereas the Air Force Academy in the upper left there and in one of our East Coast sites in the lower right there, we were dominated by target precursors. And I believe for both of these sites, they were primarily some fluorotelomere sulfonates. And then we have about 3 other sites where there's a substantial fraction of the fluorine mass is essentially present as suspect precursors. And a lot of these compounds were relatively shorter chain, maybe 4 to 6 chain zwitterionic compounds. So I think this is interesting, and perhaps it tells us a couple of things. You know, one is that, you know, despite being 10, 20 years since last AFFF application, there are still a relatively large amount of precursor compounds that are migrating in these pore waters. You know, the second thing is that if we're just performing kind of standard analysis by a commercial lab and not accounting for some of these precursors, particularly these suspect precursors, we could be missing a relatively large fraction of the fluorine that's present in these pore waters. So now let's look at pore water concentrations measured in the lysimeters, and that's the blue bars, and these are essentially the same data I was just showing a couple slides before. But now I'm comparing them to those pore water concentrations we measured in those cores we took back to the laboratory, which right now we're interpreting the latter, those laboratory cores, these red hatched symbols here, to really be more of an equilibrium number. So let's look at Air Force Academy and Peterson on the left. And generally what we see, particularly for the longer chain compounds like PFOS, we're seeing pretty good agreement there. So I think this is telling us that, at least for these sites, well, what we're seeing in the field is probably consistent with a local equilibrium assumption. We start to see a little bit of a variation with that as we get to shorter chain compounds, and I'm going to talk about that in the next couple of slides. But for this slide, let's look now to the right, and let's look at Jacksonville, at the Jacksonville site. And this is, we see something very different here. In fact, we see that the concentrations measured in the field lysimeters pretty much across the board are significantly and substantially less than what we measured in the laboratory. And what we believe the cause of this is that when we were out in the field here, and we were only down about 2 to 2.5 feet, because there was a fairly shallow water table here, so we couldn't go down very deep. But there was some heavy rainfall and a lot of infiltration. We were definitely above the water table, but there was a lot of pore water in there, and we could just tell by how readily we were extracting pore water there. And we think this rapid infiltration was leading to some sort of dilution effect, and we were seeing pore water concentrations that were much less than, say, that we would get under static conditions. So we think that this is an interesting finding, and certainly would suggest that high infiltration events might impact what we're measuring in pore water. We are going to plan to go back out there and sample again under somewhat drier conditions to see what kind of results we get. So let's talk about this a little bit, this effect of short chain versus long chain compounds. So what I have in these 3 figures is I plotted the pore water concentration that we measured in the lab, this more equilibrated value compared to what we measured in the field lysimeters. And you see for 3 sites here, and I included our Lakehurst site that I talked about initially, we see that this discrepancy, or this difference between the two, really increases with decreasing chain length. And this would, at least one way of interpreting this is this would suggest that maybe our short chain compounds, for some reason, reason are a little further away from equilibrium that in the field setting. So the question is, why would this be? So we had, for our Lakehurst site, one thing that we found is that when we did a batch kinetic desorptions test with the soil, we actually found that it was the short chain PFAS that showed very clear desorption kinetic limitations from the soils. So our first thought was, hey, maybe this is happening at multiple sites. So we ran these very simple batch tests, about a 3-to-1 liquid to solid ratio. But what we found for essentially all of these other soils, and I just have the Peterson soil here, is that we were seeing a pretty rapid equilibrium here, and we did not see some of these mass transfer effects occurring at the grain scale. So this is still a little bit of a head scratcher for us, and we're trying to figure out why we're seeing maybe this apparent effect in the field. One reason could be have to do with the flow paths in the field, and it could be that the where water is flowing and reaching our lysimeters, maybe in those zones we are pretty flushed out of the short chain compounds over time, and that's causing this effect. But this is something we're still trying to get our hands around a little bit better. Another thing we wanted to look at was potential colloid effects. There's been a couple of studies to suggest that in some unsaturated soils, PFAS absorption to natural colloids might be important, and that could be impacting our assessment of apparent aqueous concentration. So we looked at this for two of our, actually looked at this for all of our sites. I have shown results for 2 sites here. Some sites did not show any effects of this, but a couple did. So we used these batch kinetic tests, and then we analyzed PFAS, but we looked at centrifuged versus non-centrifuged samples. And we have the results of that for just showing for a couple of compounds here. Basically, for your shorter chain, less hydrophobic compounds, you don't see much of an effect. But for these 2 soils, we did see a measurable impact for PFOS. And again, we're doing some further tests to try to better understand this and evaluate this. Again, we don't see this for all soils, but we've seen it for 2 or 3 of them. One question we wanted to answer is, well, what are we potentially collecting and seeing in our lysimeters? It's one thing to see it in these kind of somewhat messy batch experiments, but are we seeing this in our lysimeters? So we repeated some of our bench scale lysimeter tests and kind of repacked the soils here, had them wet and extracted the pore water. And then with the collected lysimeter sample, we looked at centrifuged versus non-centrifuged, and when we did that, we really didn't see any substantial effects. So that tells us, at least one way of interpreting this is that any of the colloids that the PFOS was partitioning to may have been filtered out by the 1 micron opening on the lysimeter screen. So what I want to wrap up with on my last couple of slides here is talking a little bit about the phase behavior. We're going to focus on PFOS. We've looked at a lot of compounds, but PFOS sometimes is a little more interesting to look at because it is more surface active. So I have 4 of the sites shown on the left there, and then in the middle column I have the PFOS pore water concentration that we measured in the field lysimeters. The exception here is Jacksonville because of what we talked about before with the enhanced rainfall and dilution going out there. We actually used our soil pore water concentration for this. So we have those pore water concentrations, and then on the right-hand column, I have the concentration that was measured, the equilibrium concentration in those batch slurry systems. And again, the liquid to solid ratio was about 3 to 1 there. So for three of those soils, or 3 of those sites, Air Force Academy, Peterson, and Jacksonville, we see much lower concentrations in those batch experiments than what we saw in the pore water. And that can be explained just by a dilution effect. A lot of the PFAS that was absorbed onto the soil desorbed, and you see lower concentrations there. But of course, we had one soil that was a lot different, and that was our Randolph soil, and that was and and where we saw in the supernatant of this aqueous slurry concentrations that were about 35 times higher than what we measured in pore water. Now, Randolph, one of the interesting things about Randolph is it was a much finer grained soil than some of the others, and would be expected to have much higher air-water interfacial area. So we think one of the reasons why we're seeing this for PFOS is when you go into that batch slurry system, we're collapsing all of those air-water interfaces, right? So that any PFAS, any PFOS that's absorbed to that air-water interface then gets released and has to be either in the aqueous phase or the solid phase. When we looked at some of these shorter chain compounds like PFH excess, we do not see this phenomenon happening. So certainly what we're seeing here seems to be at least qualitatively consistent with release from the air-water interface. But we wanted to take that a step further and see what we could maybe infer about what might be happening at some of these air-water interfaces. So to do this, we kind of consider a mass balance approach, right? Where the total mass that you measure in these collected soil samples, it isn't just what's absorbed to the soil, but it's what's absorbed to the soil plus any mass that's in the aqueous phase plus any mass that's absorbed to the air-water interface, right? So we know what this total mass is. We know from our pore water measurements what the mass in the water is, and the concentration in the water. We can get I think a reasonable estimate of what the air-water interfacial area is based on grain size and the moisture content. And there's been a lot of work in this area. And that leaves us with trying to figure out or say something about this KAW term. And that's really the PFAS air-water partition coefficient or absorption coefficient. So what can we say about these terms based on what we've measured? Well, for the Lakehurst site, we actually did a lot of work there, and we were able, because we had so many lysimeters and a nice vacuum manifold system, we were able to collect a lot of pore water and do some air-water interfacial absorption tests. So we have a pretty good estimate of what that KAW term is, and they have units of length, so about 0.18 centimeters. Now, there's been a lot of work using interfacial tension and then combining that with various absorption isotherms, being in a Langmuir or a Freundlich, to kind of to extrapolate that based on the field concentrations and knowing what the ionic strength of the pore water is. We can get a ballpark estimate, and for Lakehurst, the Freundlich isotherm seemed to be a reasonable prediction for that. But the Langmuir didn't seem to be too far off either. For Randolph, and again, that's the soil where we saw all of this release in the batch experiment. We had a pretty high KAW there, and again, seemed to be in the ballpark of what we would predict based on the Freundlich isotherm. But what I want to focus on are the three soils I have on the bottom of this table. Based on what we measured in the total mass there and the concentrations we knew that we saw in the pore water, this KAW term was very small, and and what we would predict using a Langmuir or a Freundlich approach would be a very large overestimation. So what we're finding that, at least in some of these soils, is that PFOS absorption at the air-water interface is really much less than what's expected. And there could be a lot of reasons for this. And I think this is a pretty wide open area of research. It could be some of the other PFAS that are present in these pore waters. It could be some of the hydrocarbon surfactants or other AFFF constituents that are persisting in these source areas. It could be natural TOC. So I think there's been, a lot of interesting and fun debate about what's the most appropriate isotherm, Langmuir or Freundlich. Perhaps a bigger question for these sites is, what other constituents are there that are maybe complicating or out competing PFAS for these air-water interfaces. And I thought it was interesting in the data that Jen Guelfo showed before this, is that some of the data that was coming out of their testing showed, at least for some soils, there wasn't maybe as much impact of unsaturated conditions or air-water interfaces as one would expect in these real historically impacted AFFF soils, and maybe this is related to that. So just wrapping up, I think some of the conclusions here is that we typically are seeing pretty good repeatability among lysimeters, and I think that's encouraging for an argument for their use. We're seeing both target and suspect precursors in these pore waters, and so these suspect analytes are persisting after and potentially migrating even after many years. I think for some sites, and especially for the longer chain PFAS, we are seeing that local equilibrium might be at least a reasonable assumption, or at least a reasonable way to approximate some of these pore water concentrations. But I think there's some important caveats for that. I think, when during some of these high or rapid rain events or storm events, there might be some deviations, and in some case, some significant deviations from that. And we've seen some indications of that at some other sites as well. I think I also need to caveat the fact that most of the sites we're looking at are primarily sandy. We may have clayey sands or silty sands, but the geology is not incredibly complex, at least at the sites we've looked at so far. I think some of these equilibrium assumptions might start to further fall apart on more of the macro scale once you look at more complex geologies. And I think the impacts of air-water interfacial absorption are still unclear. I think at some sites it's been pretty clear that there's some impacts here, and that they're impacting leaching. At other sites, I think we have some indications that maybe it's not as important as we thought. And this is still very much something that we're in the process of evaluating. We have more sites for this current project that we're looking at, and I know there's a lot of groups and a lot of good people all trying to get their hands around some of these issues as well. So, why might some of this work be of benefit to the DoD? Well, I think anything that's confirming the use of lysimeters is important, as lysimeters are now being utilized at a lot of DoD sites for investigation. I think some of the things we're kind of working through and working to see here are going to serve as a basis for developing maybe more appropriate soil cleanup criteria for PFAS. And I also think some of the data that's being generated here, as well as some related projects, are really going to help in terms of developing some models and validating some PFAS leaching models that, again, there's several good folks and good groups working on. I do want to acknowledge some of the other folks who have been a big part of this project at CDM Smith, Zoom Nguyen, and Ida Fang, and at School of Mines, Professor Chris Higgins and Professor Tisa Ilangasakari, and also Stephanie Shea, who is a PhD graduate student who's been working hard on a lot of aspects of this project as well. I do want to point out two recent papers that we've published looking at field scale leaching and using lysimeters, very similar to what we've talked about in this project. And with that, I'd like to thank everyone for their attention and this opportunity to talk about this work.
Wonderful, thank you, Charles. We have a lot of questions coming in. The first one is from the U.S. Army. What is the depth to groundwater at each of the sites you worked at?
Yeah, thanks. Good question. It basically varied. I think in some cases, like Jacksonville, depth to groundwater was as little as 3 feet. For a lot of sites, we were kind of in that, maybe 6 to 6, 7, 8 foot range. And then there were a couple of sites that we were in that maybe 25 to 40 foot depth to groundwater. I should also add, and I forgot to mention this, that typically our lysimeters were around 5 feet. And of course, in some cases, if the water table was shallow, we were, we tried to stay at least a foot or so above that.
Great, thank you so much, Charles. The second question is from the Washington Department of Ecology. In your assessment, do you think that soil core transferred to the lab represents equilibrium conditions and field sampling is showing non-equilibrium conditions?
Yeah, so that was, really part of what we were trying to figure out here, and I think for several of the sites we've been to, we've gotten really good agreement between what we've measured in lysimeters and what we've measured in the laboratory on that. And in fact, it's been the exception, mainly being what we saw at Jacksonville, where we saw a big difference. Now, of course, we're still trying to confirm that. We are going to go back out to Jacksonville. So I do think, at least to some extent, we are getting in the pore water, we are getting at least a local equilibrium number in the lab, and I think, at least for the sites we've been to, I think we're also seeing that in the field.
Great, thank you. This next question is from the state of Alaska. In your opinion, and based on your work, compared to other organic contaminants, is the leachability of PFOA and PFOS in particular affected more or less by increasing the fraction of organic carbon?
Yeah, that's an excellent question, and I mean, there's certainly been a lot of other contaminants studied, so I probably can't answer that too completely. I will say that typically your longer chain compounds are the compounds that are more impacted by organic carbon and less so by the shorter chains. So maybe that's the best I can answer that question at this time.
Thank you for giving it a shot. This next question is from the Massachusetts Department of Environmental Protection. How do the organic carbon contents of the 4 soils compare, especially at Randolph with a higher KAW value?
Yeah, that's an excellent question, and I don't have those data at my fingertips right now. We certainly measured that for each of the sites. I don't think we saw very drastic differences in the organic carbon contents. So I don't know if for example, I don't know how the KOCs versus parent KDs compared for each of those sites. I don't have that data right here.
Great, thank you. And as a reminder, you can follow up with Charles. His contact information is listed here, as well as the project number is hyperlinked. You can go to the project webpage and download the final report when it's available. This next question is from the Arizona Department of Environmental Quality. Do you see any utility in using USDA soil classifications, especially the insight into oxidation and temporal saturation, to sort out the behaviors you're observing in your work?
Yeah, that's an interesting thought, and I think one of the things that we want to take a closer look at for example is what for example grain size. I think we would expect to see potentially larger impacts of the air-water interface with smaller grain sizes, just because that's going to have a higher, a greater extent of air-water interfacial area in the soil. So I think there could be something there to more closely look at, and I think that would in part explain why some of our observations for Randolph. But there also might be something to consider there when it comes to flow path heterogeneity and some potential kinetic effects we see in the field as well. So I think that is something that might be worth thinking about.
Great, thank you. This next question is from UC Berkeley. Can the chromatographic separation be linked to the soil's mineral content, for example, iron?
Yeah, so in terms of kind of comparing, for those semi-arid sites, I think it's certainly at some point the amount when you compare site to site, how they differ and also the amount of separation between compounds. Yeah, I think it is, a combination, and I think it's likely a combination of, organic matter and mineral content. But again, that might be also very related to the hydraulics, how much evapotranspiration, and maybe even some uptake at the air-water interface. So I think all of those factors might come to play.
Great, thank you so much. This next question is from the Colorado School of Mines. Do you have any information on the effects of soil disturbances, such as macropores in near-surface soils?
Yeah, that's another, and that's just kind of a classic or a traditional question when it comes to using lysimetry in the field. And there's a lot of literature, looking at nitrate and other types of pesticides, things like that are applied to the surface. We haven't looked at that very closely yet, but I guess I think one comment I can make is in that Lakehurst site, that site that I initially showed where we were had that test cell. We had tried to install some lysimeters, in the 6 inches below ground surface, and we were seeing values that seemed to jump around all over the place. So I think for those very shallow place lysimeters, I think preferential flow, macropores, all of those types of things might make interpretation of the data very difficult. I think when you get down deeper, and for us, I've been really looking at 2 to 6 feet range. At least for the soils we've looked at, we've seen less impacts of that so far.
Thank you. The questions are still coming in. Let's just do a couple and then wrap up. This next question is from Aptim. Why do you think there are such large differences in percentages of PFAS and precursors at different sites?
Yeah, that's another great question. So I think there's a couple of reasons for that. One is, and we haven't looked at this as closely as we want to yet, is kind of relating that to what we're seeing in the soil. So I think there's probably some relationship and some things we can figure out there. I think in some cases it's just the amount of partitioning that we're seeing. I think in some cases, for example, maybe some of the suspect precursors aren't being retained in the soil as much as in other sites. But I think that's an excellent question, and might even date back or transition back to the types of and extents of AFFF that were initially released.
Great, thank you. This next question is from the Naval Research Lab. Does the ionic concentration in the water matter?
So I think it does, and I think there's been a number of studies that have looked at both absorption to soils as well as uptakes at the air-water interface, where ionic strength can certainly play a big role in that. It's kind of hard to, in some extent, to quantify that site by site what we're looking at for these data. But I think I think the pore water geochemistry in general might be very important in understanding some of the phase distribution.
Great. One last question, and we're going to wrap up. For this work, is colloidal mediated transport supported for low mobility PFAS such as PFOS? And this is a question from the Massachusetts Department of Environmental Quality.
So again, that's part of what we're still trying to get our hands around. We have some batch data that suggests that colloidal partitioning and transport may be important, but then our lysimeter data says that maybe that's not happening. So I think we plan to do a few more studies on this. We have some column work from one of our other projects, and might do a few things here just to try to answer that a little more completely to see if colloidal transport is an issue.
Great. Thank you so much, Charles. Great presentation. Great job answering the questions. For the additional questions that we didn't get to, we encourage you to contact the speakers by email or by phone, or by going to their webpage and downloading the relevant documents. Our next webinar is on Thursday, March 9. It will feature DoD-funded research efforts to improve natural resource management by refining our understanding of impacts to DoD lands from disturbances and climate variability. Before we conclude, I'd like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it at this point if you can please take a moment to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you to all our audience members and to our two fantastic speakers.